Ladies and gentlemen, it's really great to welcome all of you here. It's been a few years since it was like this. So very welcome to Nobel Forum and for the announcement of this year's Nobel Prize in Physiology or Medicine. My name is Thomas Perlman. I'm the Secretary of the Nobel Assembly. I will first read the announcement in Swedish, followed by English, and we will then present the background to the prize and then open up for questions. Nobelförsamlingen vid Karolinska institutet har idag beslutat att Nobelpriset i fysiologi eller medicin år 2022 ska tilldelas Svante Pääbo för hans upptäckter rörande utdöda homoniners arvsmassa och människans evolution. The Nobel Assembly at Karolinska Institutet has today decided to award the 2022 Nobel Prize in Physiology or Medicine to Svante Pääbo for his discoveries concerning the genomes of extinct hominins and human evolution. Here he is. The laureate, Svante Pääbo, was born here in Stockholm, in Sweden, in 1955. He performed his prize-winning studies at the University of Munich in Germany and at the Max Planck Institute for Evolutionary Anthropology in Leipzig in Germany. So now I turn to Anna Wedell, a member of the Nobel Committee, who will now describe the discovery. So please, Anna.
Thank you. This year's Nobel Prize concerns human evolution. Humanity has always been intrigued by its origins. Where do we come from? And how are we related to those who came before us? What makes us different from hominins that went extinct? Before Pääbo's discoveries, knowledge was derived mainly from paleontological and archaeological findings. Bones and artifacts from extinct relatives were found at multiple sites around the world. One famous example is the Neanderthal, discovered in 1856. Many questions, however, could not be answered by analysis of ancient bones and artifacts alone. Questions such as how we and our extinct relatives are related to each other, and, importantly, what makes us different. DNA technology offered a new way of addressing these intriguing questions. DNA holds the code of life. Gradual changes to this code, mutations, form the basis of evolution, and these mutations remain in our DNA as traces from our ancestors. Our cells contain DNA in two different compartments. The long nuclear DNA carries the overwhelming majority of our genes, but the small mitochondrial DNA is present in thousands of more copies and therefore easier to study. Svante Pääbo was from an early age fascinated by ancient human history. While using DNA technology for his doctoral studies in immunology, he also attempted, as a side project, to use these modern tools for studies of ancient human remains. He soon realized that this is extremely challenging since ancient DNA is damaged by chemical modifications. It is also fragmented and present in extremely low amounts together with contamination from microorganisms and present-day humans. Svante Pääbo therefore embarked on a long series of rigorous technological developments that lasted over 3 decades. He performed much of this work on extinct animal species, but his goal was a seemingly impossible task. He aimed for our closest relative, the Neanderthal, that went extinct around 30,000 years ago. After After setting new experimental standards in this challenging area, Paabo first set out to retrieve and analyze Neanderthal mitochondrial DNA. He obtained a bone from the Neanderthal type specimen that had been discovered in 1856. From a small piece of this bone, he managed to extract and sequence mitochondrial DNA. He compared his new sequence to present-day humans and to chimpanzees and found that the new sequence was different from both of them. This was an important milestone. Paabo had proved that it is possible to retrieve and sequence DNA from 40,000-year-old bones. Mitochondrial DNA, however, contains very limited information, and Paabo's ultimate goal was to sequence the 3 billion base pair Neanderthal nuclear genome, which would allow in-depth analysis and comparisons across multiple genomic regions. The human reference genome had been sequenced in a huge international collaborative effort, and the chimpanzee reference genome sequence was also available. Sequencing the nuclear genome of an extinct hominin, however, seemed impossible. Paabo embarked on his bold plan, and for this he needed more bone. Thanks to his strong international networks, he managed to obtain 3 bone specimens from Vindia Cave in Croatia, together with additional material from Germany, Russia and Spain. All along, he refined and optimized his experimental procedures. By skillful use of new high-throughput sequencing technologies and by bringing together a large international group of collaborators for advanced computational analysis, Paabo finally achieved the impossible, sequencing and assembly of the Neanderthal genome. For the first time, important questions on our relationship with the Neanderthal could be addressed. The time of divergence could now be determined to around 800,000 years ago. Paabo and his team also surprisingly found that gene flow had occurred from Neanderthals to Homo sapiens, demonstrating that they had children together during periods of coexistence. Importantly, comparisons could now also be made to search for differences between Neanderthals and Homo sapiens, differences that may underlie features that are unique to all humans living today. Paabo's team also retrieved DNA from a minute finger bone found in Denisova Cave in the Altai Mountains in southern Siberia. Again, they started by analyzing mitochondrial DNA, and to their surprise, the sequence was different from both present-day humans and from Neanderthals. Paabo had discovered a completely new hominin entirely by analyzing and comparing genome sequences. This was a sensational discovery. The new hominin was given the name Denisova. Paabo's team next analyzed the nuclear Denisova genome sequence, enabling important investigations. Neanderthals and Denisovans were shown to be sister groups, diverging from each other around 600,000 years ago. They further found evidence of gene flow from Denisova into Melanesian Homo sapiens, indicating that mixing had occurred. Svante Paabo's groundbreaking discoveries have provided important new knowledge regarding our evolutionary history. Neanderthals and Denisovans lived in different parts of Europe and Asia for hundreds of thousands of years and adapted to these environments, while Homo sapiens evolved and lived in Africa. By mixing with them after migrating out of Africa, Homo sapiens picked up sequences that improved their chances to survive in their new environments. We now know that present-day humans in Europe and Asia can carry 1 to 2% of Neanderthal sequences, and that Asian and Southeast Asian populations carry an additional up to 6% Denisova sequences. Examples include a gene variant that improves living at high altitude in Tibetans, and sequences that affect how our immune system responds to different infections. No extinct hominin DNA has yet been sequenced from Africa, since conditions for preserving archaic DNA are poor in tropical climates. Thanks to new advanced computational methods, it has still been possible to find indications of gene flow from as yet unknown archaic hominins into Homo sapiens on the African continent. Through his persistent and innovative work, this year's Nobel laureate established a new scientific discipline, paleogenomics. This area is now expanding dramatically. After the discoveries cited here, Pääbo went on to generate genome sequences from many additional Neanderthal and Denisova individuals. In parallel, genome sequences from ancient Homo sapiens have also accumulated from different time points and different geographic locations by research groups around the world. By using Svante Pääbo's archaic genome sequences for comparative studies, important lessons are being learned regarding our evolutionary history, including how we spread across our planet. Svante Pääbo's groundbreaking discoveries allow us to address one of the most fundamental questions of all. What makes us unique? Like us, Neanderthals had big brains, they lived in groups, and they used tools, but these changed very little during hundreds of thousands of years until the Neanderthals, for unclear reasons, disappeared. Homo sapiens, on the other hand, rapidly developed complex cultures, figurative art, and advanced innovations. They crossed open waters and they spread to all parts of our planet. The basis for this dramatic development must lie in genetic changes that occurred after we separated from Neanderthals and Denisovans not so long ago. By providing genome sequences from our closest extinct relatives, Svante Pääbo has provided a novel, much more recent reference point for studies of who we are and where we came from. Thanks to his seminal discoveries, genetic variants that are shared among all now living humans, but absent in Neanderthals and Denisovans, have been identified. The list of such genetic variants is not very long. Understanding their functional implications is a focus of intense research by Svante Pääbo himself, but also by other scientists. Thank you.
So we will now open up for questions, and to our help we have Gunilla Carlsson-Hedestam, who is a professor at the Karolinska Institute and a member of the Nobel Committee. Nils-Göran Larsson, also professor at the Institute and chair of the committee, and Professor Anna Vedell, who you just heard. So please, yes. Microphone is coming.
David Keating from the Associated Press. I understand you have been able to contact this year's winner. Can you tell us a little bit about his reaction when he heard that he won this prize, and what he maybe, or what you believe this award can mean for future developments in this area?
Yes, I did reach him on the phone. This year he was situated in Leipzig, so he was not in bed. So it was easy to reach him. He was overwhelmed, he was speechless, very happy, and asked if he could tell everyone, and asked if he could tell his wife, and I said that was okay. He was incredibly thrilled about this award. I think I'll pass on the question about what this prize might mean. I'll give it to Gunilla here. Could you respond to that?
Yes, I think as we learn more and more about the genomes of current humans from different parts of the world, it's very, very important and useful to have these reference points which tell us when we diverged from these earlier human forms. And that tells us both what's unique to us, to all Homo sapiens living in the world today, but also tells us something about these gene regions that were passed on to some of the people living today from the extinct hominins. So there are a lot of implications, both in terms of understanding our evolution and with potential medical implications and a basic understanding of our physiology.
Could you elaborate on the potential medical implications, please?
Yeah, so there is, I think in regards to what makes us uniquely human, I think that a big area of interest is, of course, how we evolve to become the sort of social creature that we are in terms of collaborating in large social groups and doing this very sort of, these innovations that are very, very specific for Homo sapiens. And that has to do with brain function, of course, and other interesting aspects to study are these genes that were inherited from the extinct hominins, and they relate to many physiological functions. They relate to our immune response, as we heard, also how we survive at high altitude, many sort of interesting examples that are just being elucidated now.
Here we have one, yes.
In his research, Santi Pebo has used a lot of methods that have been developed by other scientists. What would you say is his specific contribution for which he's receiving the prize today?
Nils Göran. So his big contribution, which has been very decisive for this prize, is that he has really developed methods to analyze and recover ancient DNA. So DNA in old bones get very degraded and also chemically damaged. And on top of that, you have a heavy contamination with DNA from bacteria and contemporary humans who have handled the specimens. So he has used existing technology as this has evolved and implemented his own methods to refine the analysis of ancient DNA. So this is, of course, very important.
Could you be even more specific where he's not refining but actually finding something really new?
Yeah, I mean, I think it was certainly considered to be impossible to recover DNA from 40,000 years old bones, and you may also remember that there was a lot of erroneous papers actually describing recovery of DNA from dinosaurs and old insects in amber and so forth. And he really has worked very, very stringently with clean rooms. He has demanded that or required that experiments should be reproducible in independent laboratories. And he has also devised bioinformatic and chemical tricks to be able to analyze ancient DNA.
Any other question here? Someone.
I think it's interesting, these differences even when it comes to medical issues. And I wonder if you could be a little bit more specific when it comes to this new response. I've heard that those genes carry some disadvantage when it comes to immune response and maybe some other diseases as well.
Yes, so the sequences that we have picked up during evolution that have conferred an advantage, many of those sequences are related to immune function, but they can work in different directions. And of course today, the effects they have on us today are not immediately translatable to what it used to be prehistorically, because there were different microorganisms. So they give us a general information on sequences that are important for animal fiction. But there are also specific examples, like, for instance, COVID-19. There are sequences that confer a risk of severe disease, and others that confer slightly less risk. And in those cases, in some cases, the exact sequences aren't precisely known, but there are indications. But there are sequences working in both directions, and there are several different examples.
I know there are many who wants to get to the interviews after this press conference, but I'll still ask if there is anyone else here with a question. One more question here.
Thank you. I'm Xue Xin from Chinese Nordic Times. And my first question is, now that the Neanderthal has been intermarried with Homo sapiens, but why Homo sapiens has been survival and the Neanderthal has become extinct?
Why Homo sapiens have survived and
Yes.
why the Neanderthals? I think we do not know the answer exactly, but I think a little bit the key is that we now have a handle on that. Anyone want to elaborate, Gunilla?
I think there are several theories about that. One is that Homo sapiens basically developed very large population sizes, so the Neanderthals were sort of assimilated into Homo sapiens, and that is certainly a possibility.
Is that related to the genes, like Asian genes?
I didn't get the question, sorry.
If that's related to the Asian genes, because the genes they're toxic?
I think we can say that, as was said here, I mean, most likely there were cognitive differences. I mean, it's very hard to speculate what those cognitive differences were, but we know that there were differences in behavior, in the ability, in the group size form, the cultures that were much more elaborative in Homo sapiens, and the creation of figurative art, for example, that didn't seem to happen in Neanderthal populations. I think it's hard at this point to say more exactly what those differences are, but what we can say that they must exist, and thanks to Santiago's work, they have now been mapped and identified and are open to studies. There are already studies out that are speculating on gene differences, that are genes that are present in us, all living humans, but are different in these populations.
Yes, thank you. And the second question is, I think many people may be curious about that. mRNA technology, so I think it has been influenced a lot during this pandemic, but why this year is not recognized the Nobel Prize?
That is a very good question that I'm not going to answer. We only talk about people who are getting the Nobel Prize, and not about those who are not receiving them, or not receiving it. Yes. Okay. Is there one more question, or should we? Yes, here is one.
There was one paper that Paavo published early about a mummy from a museum in Germany, where he described the DNA. That, if I understand it right, that data was contaminated and it's no longer valid. Do you have any comment on that, or what you think about it?
It's your own.
I think that's correct. He himself says that this is not correct because you cannot get that long DNA fragments after thousands of years. So this also led him to develop procedures to be able to recover ancient DNA.
So I think we will conclude here, and then we will meet a few of you outside. Thank you very much.